genetic cross was made between homozygous wild-type males, denoted as A plus A plus B plus B plus and C plus C plus, and triple mutant females AABBCC of Drosophila melanogaster. Then the F1 males, which were denoted as heterozygous for all the loci, were backcrossed to the triple mutant females, which were mutant at all the loci, which resulted in the following F2 progenies. The order of the gene as determined from the above data was found to be BAC or alternatively CAB, and we have to find the map distance between B and C in the units of centimorgan. So let's first look at all the information that has been given. So it says that the initial parents, which are males and females, as you can see from the question, the first line says the wild type males have A plus A plus B plus B plus, so they are wild type genotype, and the females are triple mutants. So that's what we have written here, BBAACC. And remember, this is how the order of the gene has been determined, BAC, or you can also start with CAB. So you have, for the males you have B plus B plus, A plus A plus, C plus C plus, and for the females you have B, A and C. Now it also says that if you go back to the question, it says from the F1 generation, males which were heterozygote, they were picked and they were back crossed to the triple mutant females. So taking that information into account, for the next generation you have a heterozygous male which is heterozygous at all loci. So the loci are B plus AB, A plus A and C plus C, and they have been back crossed with female which is again triple mutant, so BAC. Now the question is about the distance between two genes on the chromosome. So for this, we use the information of recombination frequency. So if the genes are closer to each other, there is less chance of them being recombination event, and they are generally inherited together. So the greater the distance, the more the chances of getting a recombination event. So using that information and using the frequency of recombination, you calculate the distance between the two genes in question. So here what you see is, for the recombination to happen and for a visible change to happen, there has to be heterozygous at all the loci, which is not in the case of female, because it will just contribute BAC as the genotype. So the recombination takes place in heterozygous loci, which is present in the males. So we can just look at the possible recombination events in the male genotype and determine what could be the distance between B and C. So on this side we have written all the possible recombination events that can be due to the recombination in males. Now there is very little chance that there is recombination. Generally there are non-crossover products which are the which constitute the major proportion of the progenies. Then you have two more type of crossovers. One is single crossovers and then one is double crossover. So now, because we are talking only about three genes here, one crossover can be between B and C, B and A. The other crossover can be between A and C, and then there will be both these crossover that can take place simultaneously. So that's a crossover between B and A, and at the same time between A and C. So if that happens, these are the possible type of genotypes and the possible type of alleles that can be formed. So in the first one here, this is about a non-crossover. So there is no crossing over. This forms one allele, this forms one allele. So there are two alleles of this given genotype. And then when there is a crossover or mating with the female that can only contribute BAC, you have this kind of possible genotype for the progeny. And these, how do you identify from them from a given table like this? These will always constitute the highest number. So if you just follow the color coding here, this is dark green. So if you locate these genotypes in this given table, you should see that these should be the highest in number. So let's try to look at this. So we have to find out alleles which have ABC, and those are here, and the other will be A plus B plus C plus, which are here, and they are 311 and 317, which is the highest in the given table, and we are on the correct track. Now we move on to the first crossover event, which is between B and A. So if there's a crossover between B and A only, the possible type of genotypes are this. So B plus A C and A plus C plus. And then there is further genotype which is possible if there is interbreeding. But for this question, we only need to focus on this allele. Now, this is one type of crossover. The other type of single crossover is between A and C. If that happens, the allele forms can be B plus A plus C and B A and C plus. Now, we can also locate these alleles on the table and see how much is the frequency. But remember to put the correct allele with the correct number. So if there is this type of crossover, then the allele formation will be B plus AC and B A plus C plus. So if you locate them on these table, so B plus AC and B A plus C plus, these are first kind of single crossover event, where this can be named here as SCO1. And then if the other kind of single crossover takes place, which is the possible combination of genotypes or alleles, will be B plus A plus C. If you locate them, you find them here. And the other possible is A, B, C plus, which is this. So you have these two as the other crossover event. Now the last possible crossover is both the crossing over taking place together. If that happens, the possible alleles can be B plus A, C plus and B, A plus C. So this, if you locate, it is here 16 and 17. The other checkpoint is the double crossover event is very rare. So the products or the progenies that will be out of these double crossover events should have the least number. And in this table, as we can see, that these alleles have the least number. So we are also here on the correct track. Now we have the numbers. We can classify these progenies according to types of crossover that could have happened. So now, to find out the distance between two genes, you have to take into account all the possible crossing over that could have happened between those two genes. Now we have been given that this is the order of the genes. B and A are closer to each other, and then A and C. We just need to find out what is the distance, and that is calculated by use of the information from recombinant frequency. So let's first try with A and B. For distance between A and B, you need to take into account the crossing over that took place between A and B, the frequency of that, and the frequency of double crossover, because in double crossover also there is a crossing over between A and B. So we have to take into account this crossing over, because that was the crossing over between B and A when we accounted for. So you need to add them up. So if you just go back here, we have color coded them. We've used the same color coding here to understand. So for the first crossing over event, you have 115 plus 99, which is 0.214, and then you have double crossover events. So if you classify these crossover events according to single crossover 1, 2 and double crossover, you can individually find the frequency of those crossover events, and then for the distance between the gene, you can add these up and see what is the distance. For example, distance A and B will be this crossover which has happened between A and B and the double crossover event, so 0.214 plus 0.033. Similarly for A and C, you will account for the single crossover events which accounted for A and C, which was here, and the double crossover events that also accounted for A and C, so this and this. So this 0.247 is the distance here, 0.158 centimorgan is the distance here. So if you have to find the distance between B and C, you just need to add them up. So if you add these two up, this will become 0.405, or you can represent that as percentage as well. So if you go back to the question, it said you have to round it off to one decimal place. So when you are here, you first put it into percentage and then you put it as 40.5. So the answer to this question is 40.5. So to know more about genetics and such concepts, you can refer to this course offered by NPTEL.